But this work may have implications far beyond sticklebacks. They are convinced that there is a link between the stickleback losing its spikes and other creatures, like a manatee losing their legs. And they have two tantalizing clues. One, the same body plan gene that is responsible for the stickleback spikes also plays a role in the development of the hind limbs. The second clue is more tentative. The lake stickleback may have lost its spikes, but evolution has left behind some tiny remnants. The traces of bones. And they are lopsided, bigger on the left than on the right.
We thought, wouldn't it be amazing if in fact this classic unevenness is the signature of using the same gene to control hind limb loss in incredibly different animals.
So Kingsley and his team went looking in manatees, searching for this lopsided pattern. And they found it. In box after box of manatee skeletons, they saw pelvic bones that were bigger on the left and smaller on the right. Right now, Kingsley and his team were looking for the same switch in the manatee that caused the lake stickleback to lose its spikes. And if they find it, they will have a powerful explanation for something that baffled Darwin: how creatures like manatees, whales, and snakes can evolve away their legs. But all this begs another question. If switches can play such a profound role in the different shapes and patterns of animal bodies, from wing spots to spikes to hind legs, what is throwing those switches in the first place? Researchers would see the answer in animals very familiar to Darwin, those Galapagos finches. Arkat Abzhanov and Cliff Tabin have spent years trying to find out exactly how those Galapagos finches got their different beaks. Their starting point was what they had learned from Darwin himself. Their beaks were vital to the bird's survival. On an island where the main food was seeds, finches had short, tough beaks for cracking them open. On an island where the main food was from flowers, birds had long, pointy beaks for sucking up nectar and pollen. And they knew something else. The finches are born with their beaks fully formed. So the answer to why they had such different beaks must lie in something that happened to them as embryos in the egg.
Something amazing is happening inside those eggs. Genes are turning on and turning off. And depending on exactly how they turn on and off will determine what type of finches form.
To find out just what was going on, the researchers first had to collect some eggs.
There she is. So the female just came back. Yeah, just in the eggs. So it's very likely that she already has a clutch.
Great.
She's coming out.
Abzhanov checks a groundfinch nest and finds a single egg. He won't remove it because the mother might abandon the nest. Another nest already has 3 eggs. He takes one for his research, as he knows the mother will lay a replacement. The team collects several eggs, with embryos at different stages of development. That way they will be able to chart exactly how the different beaks grow. Back in the lab, they can begin the process. This cactus finch embryo is well on the way to its signature long, pointy beak. And this groundfinch embryo is growing a short, thick beak.
What we wanted to do was try and understand the genes that were involved in making the beak the way it was, making a big, broad, thick beak different from a long, thin beak or a short, thin beak.
They concentrated on a group of genes known to control the growth of birds' faces. As they looked, they saw something intriguing. One particular body plan gene became active in the ground finch with the short, thick beak on the 5th day of development. But it didn't go to work in the cactus finch with its long, slender beak for another 24 hours. This was a revelation. The same genes were responsible for the beaks in all types of finch. Any differences were in timing and intensity.
We've got it. We nailed it. It's the same genes in making a sharp, pointy beak or a big, broad, nut-cracking beak. What's essential, what makes the difference, and all the difference, is how much you turn a gene on, when you turn it on, when you turn it off.
And the revelations didn't end there. There was something special about this gene. Like all body plan genes, it doesn't actually make the stuff of our bodies. It didn't make the cartilage for the finch's beaks. It throws switches. And the switches then turn on or off the genes that do make the beak.
These are a different type of gene. They're genes that boss other genes around.
Scientists now realize that not all genes are created equal. Some make the stuff of our bodies, and switches are needed to turn many of these stuff genes on and off. The body plan genes are what throw these switches, which tell the stuff genes what to do and when. This subtle choreography can have profound effects on how different animal bodies are formed. And this knowledge is helping us solve perhaps the biggest Darwinian puzzle of all, the mystery of the great transformations. It all goes back to Darwin's idea of the tree of life, that all life forms are ultimately related, and from the earliest common ancestor over billions of years, they have changed and diversified so that creatures that started out looking the same evolved to become completely different. And scientists have made some amazing connections. That dinosaurs share a common ancestor with birds, and that a fish must have been the ancestor of all four-limbed creatures, even us. Of all his ideas, this was probably Darwin's most astonishing.
It was one thing to grasp how two species of finch could become different, how their beak shape could change. That was a small step. But what about the big differences? The differences, say, between the fish that swim in the sea and the animals that walk on land. How did those changes take place?
Over the years, evidence for these great transformations has been found. For instance, just a year after Darwin published On the Origin of Species, a fossil called Archaeopteryx was discovered. It had features of both birds and dinosaurs. And Darwin had seen equally persuasive evidence in embryos. Those slits in the ear of all land creatures, even humans. In us, they become tiny bones in the inner ear. But in fish, they become gills. A tantalizing hint that land animals must be descended from fish. But the stumbling block has always been how. How could a fish develop legs and walk on land? Darwin had no idea. But Neil Shubin was determined to tackle that problem.
It captured my imagination. I mean, here's a fin, and on the other side was a limb, and they looked different in many ways. And I thought, well, what a first-class scientific problem to devote my research to. And I've been devoting pretty much my research to it ever since, over 20 years.
The first stage in Shubin's quest was to find a fossil. If Darwin were right, somewhere out there, there had to be a transitional form, a fossil that was part fish, but had the beginning of legs. But where to look? He had one clue. The fossil record shows that creatures with legs first appeared some 365 million years ago. Before that, they were only fish. So summer after summer, Shubin set up camp on Ellesmere Island, just a few hundred miles from the North Pole. It has exposed rock from that crucial transitional time. The scientists' own video shows how remote and bleak the place was.
It's cold. It's about freezing every day over the summer. Winds are high. They can get up to 50 miles an hour. There are polar bears there. We have to prepare ourselves by carrying guns. It's a beautiful place. You gotta love it. It's my summer home.
Each expedition was costly, but after 3 of them, there was little to show for their efforts. A 4th trip seemed pointless.
I remember having a conversation with my colleagues, saying, well, should we go? Is this really a waste of money? This was our do-or-die moment, and we almost didn't go.
But they decided to try one last time. After 3 days, they still hadn't found anything. Then, just when no one was expecting anything to happen,
a colleague was cracking rocks, and I was working about 5 feet from him. And I hear, hey guys, what's this?
Well, look what we found. Yes.
Sticking out of the cliff was the snout of a fish. And not just any fish, a fish with a flat head. And by seeing a flat-headed fish in rocks about 375 million years old, we knew we had found what we were looking for.
a flat snout with upward staring eyes, the signature of an animal that pushes its head out of the water. And for that, it would have needed something like arms.
What we did at that moment was all jump around, high-fiving. It was there were only 6 of us in the field that time, so it was quite a scene.
Back at home, Shubin and his team got to work, examining their 375-million-year-old fossil. They named their new finding Tiktaalik, an Inuit word for a freshwater fish. Tiktaalik is a perfect transitional form. Much of its body is that of a fish. It's covered in scales. But it also had something very unfish-like. An arm-like fin, or perhaps a fin-like arm. Tiktaalik had the bone structure that is seen in the arms and legs of every 4-limbed animal. One big bone at the top, 2 bones underneath, leading to a cluster of bones in the wrist and ankle. It's the same pattern that is found in everything from sheep to sheepdogs, to Shubin himself.
You now have an animal that can push itself up off the substrate, either on the water bottom or on land.
One obvious question was, why had Tiktaalik evolved this new structure? One possible answer is suggested